उधर पे
Hello, students. Very good evening. Welcome back to the same sessions of our MBBT. Uh, I hope you are all doing had a good life till now, and also in future, safe life. That's what we require for. So there are some questions I can see that somebody wants is in the last year biotechnology. 1 year bioinformatics, then apply for masters. Why not now? You can apply for masters in Utrecht University. So it doesn't matter if you have a bioinformatics diploma or not. They just your life will start from your masters. Diploma will be there. If your masters in bioinformatics, then that might be helpful. But you for sure you can get admission in Utrecht University based on your bachelor's degree. So don't waste 1 year in doing diploma in India. Maybe it's a you can save 1 year by studying their some other course, their languages maybe. I would really suggest. Dutch is the language in Netherlands they speak, so you can learn that language instead of having a 1 year bioinformatics because that you will learn if you are going there. So my email ID is this one. So yesterday we did some biotechnology techniques. Oh yes, you mean stem cell culture, regenerative medicine. It has a huge scope, very huge scope. I was working in stem cell culture for a couple of months, and they were creating organ plants like heart samples via stem cells in a batch. So it was so that was my group. We were studying there. So they didn't put up anything at the moment. So there are various groups that are working. So you can check various groups. These are the people who are working with the regenerative medicine very well. So just look, or you can look at the DAD website for further information. There's a lot of information available on that site. Lot of cookies. So just go through the website. You can have plenty of information from them also. No, just call them. You can call them from here. There will be some numbers of them, or just contact on their Facebook group or something. Any student on the Facebook, or who could contact you, the secretary of the professors who is dealing with that course. Just directly call them and ask them that this is my problem. Will I be eligible for this? And get your answers directly. Do a lot of online courses, Coursera, like what you're doing now. Do as many as courses. Have your uh, its main thing is not, it's always not like getting certificates and having internships. It's all about learning out of them. That whenever you are on interview, you are bringing those certificates with you, not showing them as a piece of paper, or on your resume. Nobody is interested in that. They're all interested in your knowledge. That whatever the certificates that you have earned, that they have some meaning and you know about it. So somebody asks you about molecular biology and biochemistry techniques, you did that. So you can say some of the techniques from our course, that I did these techniques. So I had some virtual experience also, but didn't have practical experience because of pandemic. But I still, if I'm allowed to do so, I can do it. So that was the main idea of this course, that you are standing in front of person, you can explain these techniques, you have learned them, you are heard of them, and you are just in need of a place where you can work and start working. That's all they need, not much of it. Oh yes, CRS, clinical research associate. So after BTech, you can go for 1 year diploma course in clinical research associate, where you can get as a job as CRA afterwards, and it's handsomely paid, and it could have its own, yeah, it could have its own advantages in the CRA field. So go for that. Medical genetics. I'm not a genetics guy, so I'm not sure about that. Okay. Hello, hello. No, I will not suggest going for medical coding. Just stick to clinical research associate program, or pharmacovigilance is also a hot thing. Do something related to that. Something with pharmacovigilance, clinical research associate, which could have good money also. Medical coding has a future, but it doesn't. It's I didn't saw a big future afterwards. Your career will be stagnant after some time. So, bache, if you want to look for good universities in Canada, I would suggest um, check online. So, McGill is number one. British Columbia is good. University of Toronto. So these are the few names. Few names. that could be. Yeah, University of British Columbia, BC, Toronto, McGill, many more, many more. It's a huge world there. So let's start for today's work. So today I will be dealing with the, oops, why is it at the end? Very short slides about fluorescence activated cell sorting. So we will be dealing with the fluorescence activated cell sorting, how this technique is important, how you can sort your cells like RBCs, WCs, disease cells, non-disease cells with this technique, and get to know about real-time data with the help of this technique. So we will basically deal with the basics of flow cytometry, understand how flow cytometry sorter works, understand how cells are sorted, description of different cell sorting techniques, tips on sample preparations, and overview of sorting services by a company. So which techniques are appropriate for my cell separation? So whenever you are going for sorting your sample, so there could be two types, either they could be tissues or fluid particles in suspension. So the ones in the tissues, they could go for laser micro dissection microscopy. The one there which are in the fluid form, they could go for FACS, microfluidics, or large particle sorting like COPAS. So there are three techniques for the same. So majorly today we will discuss about FACS, that is flow cytometry technique. And what is flow cytometry actually? Flow means fluid, cyto means cells, metry means measurement. So measurement of cells which are in fluid. So that's how you define flow cytometry. So you check the cells which are in the fluid and measure them actually. So what types of particles can be analyzed? They could range from cells, chromosomes, bacteria, yeast, microparticles. So all these ranges are possible with the help of flow cytometer. And what particles can be analyzed? They could be range from 0.5 micrometer to 14 micrometer. So bacteria is 0.5. So phytoplanktons, red blood cells, CBABs, lymphocytes, neutrophils, monocytes, and even further. So they all could be analyzed with the help of flow cytometer. So three key parameters that are measured in this flow cytometer are the relative size, the size of your cells, inner complexity, how your cell is complex, and a relative fluorescence intensity, and how much intensity the fluorescence they are emitting after the immersion of electron beam to it. So these are the various sample runs analyzers. That's how a flow cytometry looks like from various companies. Mainly they are from the BD Biosciences. So sorter, so they are in flow cytometer, they are sorter are available, which could be divided into three main subsystems, that is fluidics, optics, and electronics. Fluidics department deals with the introduction and focus of the cells for interrogations and create a stable break-off for sorting, whereas optics deal with the to generate the collect the light signals, whereas electronics deal with the convert the optical signals to proportional digital signals, process the signals, and communicate with the computer. So three different paths, they're working together in the sorter system. So how do we record them? The sample is first introduced into the running sheath fluid in the flow cell. The sample is introduced in the flow sheath of a flow cell. Then hydrodynamically they focus in the core stream. So hydrodynamically they are focused in the core stream in the center. Sheath fluid and samples do not mix. The sheath fluid is outside fluid and it does not mix with the sample. And reduction of cross sections causes an acceleration. So cross section is reduced, which leads to the acceleration of your samples to the laminar fluidics. And sample rate flow could be adjustable. So rate could be 10 microliter per minute to 60 microliter per minute, being respectively high or low and high. So that's how your cells are moving in the center in the case of fluidics department. Then in the optics, after the fluidics is done, your cells are flowed away with the acceleration. Now comes the part of your lasers and fiber optic cables that carry beam to steering prisms, which can direct laser beams to the stream. So after the lasers are running through your samples, they've gone through some prisms with the help of fiber optic cables. They can directly to the stream. Then they could go to the flow cells like this. So these are on the left side fiber optics, and these are the X-mount optical plates are placed, which could lead to the focus, and prisms are making it more precisely. It could lead to your cells then. And there are two different type of optics systems in sorter: fiber optics and photon multiplier tube. Fiber optics directly emitted light to the appropriate emission filter block, and PMT, they direct the signals to emission block to appropriate photon multiplier tube. So there are different types of arrangements of filters. They could be trigon, like 3 times A, B, C, and they could be octagon. So A, B, C, D, E, F, G. So all these are possibles. Then photon multiple PMT. What does PMT looks like? In this case, photon light is focused through cathode, and there's a vacuum tube inside in which the multiple citation is happening, then leading to the very large volume of your samples. And then anode, then they are getting results are getting absorbed at anode. Then so how are cells actually sorted? So to sort your cells, so this is the first step. This is the machine that you can see very nicely. Samples generate light scatters. Here are samples generate a light scatters and fluorescent signals, and signals are analyzed. After the sample is done, then the sample has they move into 3 parts. So charge is also applied via stream charging on top. Then charge droplets break off. So because of the charge droplets break off into 3 different parts. One is the neutral in the center, and left is a one charge part, and right is a different charge part, positive and negative. And in the center is the deflection plates attracts or repel charged droplets. Then on the fifth is the uncharged particles pass to the waste. We will just throw them away. And on the other hand, the charged droplets containing the particular particles of interest are collected. So whatever the interest of particles are, we can collect them. So here you can see how your droplets were coming out. Then there's a break off of your droplets. They were in the single stream. Like here you can see live image of the stream taken from the video monitor. Drops are visible because stream is illuminated by a strobo lamp, which is driven by the drop drive frequency. So we are seeing with the camera. So initially they are all in drops, then suddenly there drops off, there's a break off, and then they are changed into the negative and positive sides, and they're sorted as a green and orange like this. So here the processed image from the video capture card. Notice the ligament extending from the connected and minor fast which melt and main drops on their way down. So into what collection device can I sort my samples? So you can sort your samples in bulk. Sorts are done with the purpose of enrichment of cell culture, DNA RNA analysis, adaptive transfer, purification of cells expressing a fluorescent protein. And cell can be sorted into 1.5 ml per tubes, 5 ml Fox tubes, 15 ml tubes, beaker. Samples could be cooled down or uncooled down. So you can do in various ways your cell sorting. You can also do in the plates. Plate sorters are available. They are done with the objective of sorting directly into the multi-well plates for the purpose of tissue culturing, T and B cells clonal expansion, single cells DNA and RNA analysis. Cells can be sorted into 6 to 384 well plates in that case. Then there is a slide sorts, like in the slides we can sort them. They are done in the purpose for in vitro assays or microscopy. So some quick tips on sample preparation. So plan your experiment well before. Choose fluorescent antibodies that you are looking for. Agree with the optical centrifugation of intended sorter. Note that different sorters may slightly vary in optical configurations. Bring along following controls: unstained transfected cells, some single stain samples, and FMOs to set the gates. Maximum of 2% of FCS need to be there in the medium, otherwise cells will be very sticky to each other. Avoid phenol red media due to autofluorescence. Most cells types will not do just in PBS or FCS. If your cells sense to aggregate, consider adding one of the following: use calcium or add EDTA, or you can add cell lysis with DNase 1 MgCl2, or you can also add acutase in sorting buffer. So all samples need to be filtered, preferably directly before sorting. Before sorting, filter your samples via nozzles depending upon that. So 70 microliter nozzle you can use, 85, 100 according to the cells number of cells you have present. So minimum sample volume for sorting is 200 microliter. Volumes can be loaded from 1.5 ml Eppendorf, 5 ml Fox tubes and 15 ml Falcon tubes. So choice of collection depends upon the downstream application, is therefore variable. So harmful substances like 2-mercaptolone, triazoles are not allowed into the cell sorter. So if you can measure it in our analyzer, we can sort it our sorters. So possibly to sort the smallest bacteria to largest adipocytes. So plan your fluorochrome panel according to the optical quantification of sorter to be used. Prepare your samples following the guidelines of bulk versus single sort. Do not forget to use controls and bring appropriate collection media for downstream technique. Thank you so much on the behalf of Edufabrica team for having this little workshop of today. So now I will take you to some videos which will explain more these things into more scientifically, I would say, and more easy to make it more clear. So it will be divided into two parts of 10 minutes. So just grab a popcorn and then we start this.
Hello guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually, I did two videos, so in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell passes through the laser beam, there are two events happening. First is the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC or a data analysis analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now I'm going to speak more in details. Saying that this is the flow cytometer tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak, like high voltage peak for large cells and low voltage peaks for small cells. The data, or the forward scattered light data at the end, will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction, Yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of the light into voltage, and then I'll get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells, because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes, because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about Fax mechanism. In the next video, I'll speak about Fax mechanism. So if you want to know more about Fax mechanism, then you need to watch the next video. If you like this video, please don't forget to like. Hello guys, this is a continuation of the subject flow cytometer and Fax mechanism. In the previous video, I spoke in details about flow cytometer, and in this video, I'm going to speak about Fax mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand Fax mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about Fax. Fax mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. Fax mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A fax machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture, and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells, the cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now, when the cell pass through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore pass through the laser beam, the fluorophore will be excited. And then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So, for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scatter light. When the cell appears here, it means that these cells are small cells because they have low signal on the forward scatter and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appears here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scattered light. Then when the cell appears here, it means that it's expressing the protein, but it's a small cell. And when the cell appears on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescence cell with positive charge, for example, and the low fluorescence cells with negative charge. I can choose actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescence cells with a positive charge, and the big low fluorescence cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them. So it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if the cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, then or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with the protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal of on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with pHC. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector or on pHC detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about that, as you know, now we can in the new fax machines, we can analyze like tens of proteins and tens of cells in one time, because new fax machines have like some machines like there is now one fax machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel. If you have any questions or any suggestions for other topics, write me in the comments, and see you.
So students, I hope it was a long videos continuous, but my reason was to teach you via this videos that you can have various as she has explained, that fluorophore 1, fluorophore 2, for 2 we can differentiate between fluorophore 1 and fluorophore 2, and we can also differentiate between healthy cells and normal cells actually. Then we can check whether which antibody has been attached to it, and we can sort our cells and according to that. So in this case we have CD14 protein cells and CD4 protein cells. So this is how these things have explained everything in very clear-cut manner. So last but not the least, we'll take you a 3D image of this, just 2 minutes video. Then we go for virtual lab also. If you wish to study about cancer cells and normal cells, how we separate them from normal cells from a tissue. I mean, how you can separate. So I mean, flow cytometry is one of the technique which could help you students to separate them, and you can have cancerous cells on one histogram and one on the other side the normal cells, and it will differentiate based on the fluorophore, and then you can have the results out of it later on. Thank you. I know. So students, it's almost 57. I meet you at 5. Then we go for 5:05. We go for this flow cytometry. Uh, Small workshop about that. So, so blood is made up of a lot of things.
Select a blood cell. Red blood cells carry oxygen from our lungs to all of the body's tissues, and they carry carbon dioxide away. White blood cells help us to fight infections. There are multiple types of white blood cells, each with a different specialty. When we have a cut, platelets help stop the bleeding by allowing blood to form a clot.
Platelets, RBCs, and WBCs.
In a healthy person, each type of blood cell is present in predictable proportions. If the proportions fall outside of what is considered normal, it can be a sign that something is wrong.
So something goes wrong between RBCs, platelet, and white blood cells. When we have a blood test, we see that something is wrong with our blood test, and then things goes completely different.
A complete blood count, or CBC, is one of the most commonly ordered lab tests. The test measures the proportions of different types of cells. CBC tests used to be done manually by looking at blood through a microscope. In a few minutes, a technician can count about 100 to 200 cells. Let's see how many you can count in a minute.
An automated blood analyzer, on the other hand, can count many thousands of cells in just a few seconds. Its speed and accuracy make it a much more popular choice. The machine draws in a small amount of blood. Red blood cells and platelets have distinctive characteristics, so they can be counted with a method based on electricity. The differences between white blood cells are more subtle. To tell them apart, the analyzer isolates them and then uses flow cytometry to count them. The cells pass through a narrow tube in single file. One at a time, they pass by a laser. When the light hits the cell, it scatters. Across from the laser, a detector senses the amount of forward light scatter. Larger cells cause more forward scatter, and smaller cells cause less. Another detector senses the amount of side light scatter. Cells with a lot of internal complexity cause more side scatter, and simpler cells cause less. Computer software counts the cells and plots their light scattering properties on a graph. Since each cell type has a different scatter profile, we can use the graph to identify the cell types present in the blood sample and their proportions. Does this person's blood count look normal? Compare these results with those from a healthy person and note the differences. The plot doesn't look quite right. This person has many more immature white blood cells in their blood than normal. To get a better sense of what's going on, take a look at the blood under the microscope. The results suggest acute leukemia, a type of blood cancer. The patient's doctor will follow up to see if these results line up with any symptoms the patient may be experiencing. Leukemia happens when a stem cell in the bone marrow starts to divide out of control. Bone marrow is where most of the cells in our blood come from. In a healthy person, stem cells there divide to make new cells, which then mature and move into the blood. In the acute type of leukemia, immature white blood cells grow too quickly and fail to mature. They soon crowd out the other cell types. To stop the leukemia cells from growing, it is important to begin treatment right away. But the right treatment depends on identifying which type of cell is growing out of control. The CBC results from our patient suggest that they could have either B-cell or T-cell leukemia, but we don't know which. These cell types look the same under a microscope, yet they respond very differently to treatments. To find out which cell type is growing abnormally in our patient, we will need to analyze a sample of their bone marrow. Add some lysis solution. This step breaks open certain types of cells and leaves the ones that we're interested in intact. Next, we'll add some tags. The tags recognize specific molecules on the surface of the cells. The molecules themselves are too tiny to be seen, but the tags will allow them to be differentiated. The blue tag attaches to a molecule on the surface of B cells. The orange tag attaches to a molecule on the surface of T cells. Flow cytometry is a tool that can be used to get information about your blood. In this virtual lab, you will use flow cytometry to do a complete blood count, diagnose a type of leukemia, a blood cancer, and look for any signs of cancer that remain after treatment. After 4 weeks of treatment, flow cytometry is used once again to look for cancer cells that remain in the bone marrow. This testing is important because if cancer cells are still detectable, we'll want to give the patient a more intensive therapy. If there are no detectable cancer cells, then we know the treatment is working, and we will continue with it. Flow cytometry can analyze many cells very quickly, and it can detect even one abnormal cell among 10,000 healthy ones. This makes it the most sensitive and accurate measure available. It looks like treatment for our patient is going well. Back in the 1960s, B-cell ALL was deadly about 95% of the time. But today, thanks to flow cytometry and improvements in cancer care, 95% of patients with this diagnosis will recover.
So students, so that's the sessions for today. I want to do gel electrophoresis, but maybe we do it tomorrow. And so I will share everything for this today's workshop. All the videos. One video I didn't play, that will be too much for you, so I just keep it simple. So happy learning. Is it possible to separate viruses of different serotypes by this type of method? Um, Maybe it's possible because of their different structures. We can differentiate them. So mainly it's sorting every cells of different structure type also based on their complexity, and also on the based on their fluorescence technique that we used based on the protein that they are having. So maybe it's possible with that also. So further questions, students? If not, I would like to end the session of today. So you will get the documents. It's being uploaded. Thank you very much. It was wonderful with you all. Bueno nachos. Yo, bye bye. Take care. Have a nice time with your family. And yeah, take care of your health, near and dear ones. Danke schön. Bye bye.